Good afternoon, students. Today we're going to do lab number 10, which is the lab on DNA and molecular biology. And our first exercise, exercise A, we're going to isolate DNA from strawberries. Now, why strawberries? Because they give a good output of DNA compared to other, fruits and vegetables. We've tried onions and peas and they work good too, but strawberries are the best. So we're going to take the strawberries and mix it with some water and some sodium chloride salt. And then we're going to add that to a blender and homogenize for a few seconds until you got a nice pulpy homogenate. So once we've blended the strawberry pulp, we're going to filter it through cheesecloth, collect what comes out of the cheesecloth, and we're also going to mix the strawberry pulp, I guess you'd say, with a detergent, right? With the detergent helps to homogenize and separate out the nuclear envelope, the cell membranes, and even cell walls if we're doing other plants, right? And then we're also going to add to that Adolf meat tenderizer. What the meat tenderizer does, it separates the histone proteins from the DNA. If you remember, eukaryotic DNA is wrapped around histones in a beads on a string kind of arrangement. So we want to unwrap that, take away the protein so that all we have is DNA. Once we have that, we're going to precipitate it with 95% cold ethanol. What that's going to do is make a phase so that the DNA will be kind of in the phase between the bottom, I'm going to lift it, between the bottom of the tube and the top, and you'll be able to collect the DNA. Okay, so once you've added the cold ethanol to the tube with the strawberry extract, you wait for a period of time, 20 minutes, 30 minutes, depending on what the protocol says, and you'll be able to see that the DNA will come out of solution and you'll be able to see some snotty strands of DNA inside the tube. We'll take a wooden stick. You could also use a glass rod that's been scratched, right? And you're going to spool the DNA out so that you'll be able to see how long and thin the DNA is, and actually quite snotty. Once you take it out, you'll be able to visualize it. If you were in a biotech lab, you take the extracted DNA and then do other things with it. You might run it on a gel, clean it up some more. This is what we call a down and dirty protocol for DNA extraction that does not involve any dangerous chemicals because, after all, we don't have that in this lab.